My dear students of 12th class, today in your botany we are going to take up the inheritance of one gene as carried out by the father of genetics, that is none other than Gregor John Mendel. And previously we have discussed about the 7 pairs of contrasting character traits in pea plants which were studied by Mendel. If you remember, these are the seed shape and the round or wrinkled, seed color yellow or green, flower color violet or white, pod shape full or constricted, pod color green or yellow, flower position axial or terminal, and then the stem height either tall or dwarf. And now we will move ahead and try to study the inheritance of one gene as carried out by Mendel. The hybridization experiment carried out by Mendel, in which he crossed tall and dwarf pea plants to study the inheritance of one gene. For this inheritance of one gene, Mendel chose the tall and dwarf pea plants. He collected the seeds produced as a result of this cross. That is, he performed a cross, though you must be knowing that the crosses he made on huge populations to study a particular trait or character. So he collected the seeds produced as a result of the cross made between the tall and the dwarf pea plants and grew them to generate the plants of the first hybrid generation, and this generation is also called the filial progeny or the F1 generation. Now, Mendel observed that all the F1 progeny plants were tall, like the one of its parents. None were dwarf. Now you must try to understand this. He crossed tall with the dwarf, and the result was the F1 generation, in which all the plants that were produced, they were tall plants. All plants were tall. And then Mendel observed that all the F1 progeny plants were tall, like the one of its parents. None were dwarf. And Mendel made similar observations with other traits, pairs of traits, and found that this was to confirm whether this will be true for other traits also. And Mendel made similar observations when with other traits, pairs of traits, and found that the F1 always resembled one of the parents, and that the trait of the other parent was not seen. And still he didn't make any conclusion. Then Mendel then self-pollinated the tall F1 plants, and to his surprise he found that in filial 2 generation, that is F2 generation, some of the offsprings were dwarf. The character that was not seen in the F1 generation was now expressed. You can visualize here that when all tall plants were subjected to selfing, that they produced the seeds, and the same seeds were sown in the F2 generation. The plants were produced in the ratio of 3:1, with 3 of them being tall and 1 dwarf. This was the F2 generation, and that was produced from by selfing the F1 generation, all tall plants. And in F2 generation, the ratio was 3:1. The character that was not, or the trait rather, that was not expressed in the F1 generation was now being expressed in the F2 generation in the ratios of with 3:1, that is, 3 were tall and 1 dwarf. Though his experiments were of large sampling size, and finally he observed that the ratio of tall and dwarf was in 3:1 ratio. And then Mendel then self-pollinated the tall F1 plants, and to his surprise found that in the filial 2 generation some of the offsprings were dwarf. The character that was not seen in the F1 generation was now expressed. And the proportion of the plants that were dwarf were 1/4 of the F2 plants. Dwarf were 1/4 of the F2 plants. That means out of 4, 1 plant was dwarf and of the F2 plant, while 3/4 of the F2 plants were tall. Hope you understand this. I may repeat this. The proportion of the plants that were dwarf were 1/4 of the F2 plants, and 3/4 of the F2 plants were tall. The tall and the dwarf plants were identical to their parental type. This is to be mentioned here. The tall and the dwarf plants were identical to their parental type and did not show any blending. There was no blending. That is, the offsprings were either tall, similar to their tall parents, and either dwarf, similar to the dwarf parent. None were of in-between height. And similar results were obtained with other traits that he studied. Only one of the parental trait was expressed in the filial 1 generation, while at the F2 stage, both the traits were expressed in the proportion of 3:1. The contrasting traits did not show any blending at either F1 or F2 stage. Now, from the above observations, Mendel proposed that something was being stably passed down unchanged from parent to the offspring through gametes over successive generations. He called these things as factors. Now we call these factors as genes. Genes, therefore, are the units of inheritance, and they contain the information required to express a trait, a particular trait. So, Mendel's factors are now known by the popular term that is the gene. Thanks. Have a nice day.